Diabetes treatment, bridging the divide. That is the heading of the topic. Underlying is the glucolipotoxicity. So I correct the glucolipotoxicity and bridge the divide. What is the divide? The divide is on one side is the effort to increase the insulin secretion from the beta cells by giving uh, secretagogues or drugs to increase the peripheral utilization, the present mechanism. The present mechanism is treatment oriented when there is a problem. The future mechanism, which will be to prevent that itself and then prevent glucolipotoxicity and see that the beta cells are forever secreting adequate insulin without apoptosis. That is on the other side. So how do we bridge from this to that? Approach to treatment is twofold. One, a treatment oriented when there is a deficiency of insulin, try bring more insulin, either by beta cell stimulation or give exogenous insulin, or use the existing insulin by sort of insulin sensitizers like your metformin or any one of the other drugs which are insulin sensitizers, or increase the secretion of insulin through your DPP4s and GLP-1s, and all that is one story. The other story is, how can I prevent the beta cell from apoptosis? How can I see that the beta cells are forever healthy and then secrete insulin and then not produce diabetes? It's both preventive and curative. That is the basis of this talk. Tamasoma Jyotirgamaya, from darkness to illumination, rightly sets the tone for this talk. You see here, that's the pancreas there, all of you know. There is pancreatic duct and the SNI are there. And each the SNR duct and the SNR cells, these pancreatic SNR cells are surrounding a group of beautifully colored endocrine cells. And this is called these are the pancreatic endocrine pancreas. This is all the exocrine pancreas. So in the endocrine pancreas, I have 4 different cells. One, the alpha cells, which secrete glucagon. The other one, the beta cells, which secrete insulin. And then you have the delta cells. And then these are, of course, islet cell of Langerhans, pancreatic SNI, and the PP cells, and of course, the blood vessels there with the red cells there. Alpha cells, glucagon, beta cells, insulin, delta cells secrete somatostatin, PP cells secrete PPY and NPY. Okay, these are the secretions. Most of you may be aware only about these two, and keep a note that the others are gaining a lot of importance as far as the architecture of pancreas and its secretions. Now, very simply, to understand the biochemistry, there is glycogen in the liver, and that glycogenolysis or glycogen degradation, glucose 6-phosphate is the central molecule, enzyme, which makes glycogen available as glucose and again puts glycogen in the stores whenever there is excess of glucose in the body. So what is the central molecule for carbohydrate metabolism?
Glycogen.
Glucose 6-phosphate. Glucose 6-phosphate is the central enzyme, and that glucose 6-phosphate works both ways. It puts the glucose into glycogen and brings glycogen back into, and this 6-phosphate, once the glucose is there, glucogenesis and glycolysis. Gluconeogenesis is this side and glycolysis is this side. Now, ATP is released by that, and then the pyruvate comes, and pyruvate enters the Krebs-citric acid cycle. Acetyl coenzyme E is generated, and acetyl coenzyme E can become ketone bodies and get metabolized or enter the Krebs-citric acid cycle, oxaloacetic acid, and then again ATP is released here, and this is the mechanism in which glucose is metabolized. Go to triacyl glycerol. Triacyl glycerol, this is triglyceride, and then the glycerol moiety is removed, and you have the free fatty acid. That free fatty acid is available for beta oxidation. And there's also synthesis of free fatty acid from ATP mediated acetyl coenzyme A can either go down and to produce energy or can synthesize fat depending upon the requirement. Oxidation is downward pathway and synthesis is upward pathway. And again, the acetyl coenzyme A is the central molecule. The central enzyme is glucose 6-phosphate and central molecule in the metabolism is acetyl coenzyme. It has got three routes. One can go into carbon dioxide and energy through the citric acid cycle, or it can go through the ketone body pathway and get metabolized, or it can go and synthesize more fatty acids and more fat formation. Okay. Now what happens when you have excess energy here coming down here, more fat can form. Okay. Or if you have more fat, it can get degraded and give more acetyl coenzyme A, which can get into ketone bodies, and the excess acetyl coenzyme A may prevent this pathway of glucose metabolism. So both ways, inhibitory as well as facilitatory. And then coming to the third important molecule, the energy molecule is the protein. There are amino acids broken down from the protein amino acid degradation and amino acid synthesis. And these amino acids can increase the pyruvate. They can enter into the acetyl coenzyme A, or they can enter here also, or they can get metabolized into urea. So this is nutshell the metabolism of three basic foods: carbohydrates, fats, and proteins. So the intermediary metabolism, as I told you, G6P is the central molecule. It can go to glucose, it can go to glycogen, it can go to ribose 5-phosphate, it can go to acetyl coenzyme, depending upon the requirement. This acetyl coenzyme can generate energy and carbon dioxide. This can lead to phospholipids. This can lead to cholesterol synthesis. This can lead to fatty acids. The primary cholesterol also is synthesized from acetyl coenzyme. Needless to say, G6P and acetyl coenzyme form the main route through which everything is channeled. Whether G6P will be glucose 6-phosphate will be converted into glucose or glycogen or ribose 5-phosphate or acetyl coenzyme, depending upon the energy requirements in the cell, it manipulates. But in disease, what happens? Several problems occur. The G6P to function, the glucose to enter into the cell, they require a passport, and that passport is the insulin. So you can see here there are different mechanisms through which molecules move across the cell membrane. The colored one here, this is the cell membrane. You start with, substance is in and out. That is the facilitated diffusion. Primary active transport, ATP induced transport. And this is called active transport. One ion gets in, the other ion gets out. And this is called the exchange transport, ion exchange channels, ionophoric mediated ion transport, where there is energy dependent, and then a simple diffusion. So starting from simple diffusion, facilitated diffusion, primary active transport, and then exchange transport, importer, and then the ionophore mediated. 6 different ways the molecules move across the cell membrane in various scenarios. So there are several transporters here, and we are interested to know in our presentation what are called the SIM transporters, which is glucose and then sodium SIM porter or SIM transporter, co-transport. They will be together transported, SGLTs, and also the gluts. So you need to know about the glut, GLUT. Glut is glucose transporter. Short form for glucose transporter is glut. So these are again uniporter, only one direction, and the symport is bidirectional. One molecule gets in, another molecule gets out. Antiport is, this again, the other one molecule gets in and the other molecule is secreted out. Symporter both get in. Along with that, this is the glucose SGLT is the symporter. SGLT is the symporter, and the antiport is one gets in, usually potassium. Hydrogen or potassium sodium exchange is the antiport. So cell membrane different transporters. Now coming to the glut, GLUT, you just imagine a clip that you hang the clothes. It will be something like that. So if you press it, the one end will open. If you don't press it, that end is closed. So something like that. It is like a double hanging clip. It is like that. So when the glucose arrives there, it has to get open, but of course it is closed here, and it has to go down. So the glucose molecule comes here, and then this end gets open, and the glucose is squeezed out, and again it is ready to receive another glucose molecule. So there are how many gluts are there now? 13 gluts are there, or 14 gluts are there. So you don't have to remember all the things. What we are interested is the GLUT2 uniporter of glucose into the beta cell and stimulates insulin secretion. So this is what is called glucose mediated insulin secretion, or GIGSIS. Okay, beta cells of pancreas, liver, small intestine, epithelium, kidney has a high kilodalton, 60 millimoles, macromoles, and then never saturates. Glut can never be saturated. So this is the very sort of abundant receptor we have, GLUT2, all over the body. Most important, GLUT4 is insulin dependent. For this channel to work, insulin is required. For GLUT2 to work, insulin is not required because it is insulin producing, and GLUT is there, is most profoundly there on the beta cells. GLUT4 is there on all the tissues, where for the glucose to enter, it has to come with the insulin. Whereas in GLUT2, for the insulin to come out, we are using glucose there. Glucose induced insulin stimulation there. Here, insulin induced glucose movement through GLUT4. So, okay. So let's move faster a bit so that we have to cover several things. And yeah, this Rajaram has shown. What happens is when the glucose enters into the cell through the GLUT2, and then ATP is produced, and this ATP ADP conversion, energy is released. ATP is broken down to ADP, and phosphate energy is released. That phosphate energy closes the potassium channels. Once the potassium channels are closed, voltage dependent gated channels of calcium are open. Calcium influx sectors. The calcium influx coupled with the intracellular mechanisms, there will be an increase in the delta calcium. That is, the balance of free calcium versus the bound calcium. That produces exocytosis of the insulin granules and insulin secretion. In nutshell, glucose comes to the beta cell through the GLUT2, it enters inside, produces ATP. ATP becomes ADP and produces phosphate. Phosphate closes potassium channels, and then closing of the potassium channels automatically opens the calcium channels. These are the voltage dependent calcium channels, and they are open. Calcium influx. Calcium, if it is more intracytosolic calcium, is more. Already preformed granules are there in the insulin granules, are in the granular form in the endoplasmic reticulum, and they are stimulated to get released and insulin release. GLUT enters beta cells through uniportal GLUT2. This does not require insulin because its very purpose is to produce insulin. Oxidative phosphorylation. ATP closes the ATP gated potassium channels. Depolarization opens the calcium channels. Calcium enters into beta cell. This leads to exocytosis of insulin and secretion. Remember, this is the way insulin is produced at the beta cell. Okay. Now, this is an intricacies of that are shown here, nothing very much different. So through the GLUT2, the insulin, the glucose has come. Glucokinase is acting there, glucose 6-phosphate. Glycolysis occurring in the process. ATP is generated, that ATP sort of depolarizes, and then potassium channels are closed and calcium channels are open. Calcium comes in, diacetylglycerol, 1,4,5 ionocetyl pyrophosphate 3 will form. Calcium sensitization releases the intracellular stores through the PKC. Exocytosis occurs. These are the granules, and then exocytosis from the endoplasmic reticulum, whatever is synthesized and then produced in the granules there, and that they become open and then come out. So in the insulin synthesis, there is a gene which synthesizes the insulin through these mechanisms: PX6, NeuroD, PDX1, E2A, and these transcribe and produce the insulin amino acids, and then splicing. that is folding. This folded one enters the endoplasmic reticulum, and that is stored in the insulin granules, and that is released through this mechanism. Okay, so that is the one which we have already discussed. Now, the secreted insulin is like this: the proinsulin, pre-proinsulin, and the endoplasmic reticulum. The pre-proinsulin is now sort of is spliced up and then cut off here. This is the cut off here, and when there is a scissoring off, this remains, and that is called the C peptide, which you measure. It's in the C shape, and this is the insulin. It has got alpha chain and beta chain. So the beta chain and the alpha chain with the long amino acids there, the single peptide in the endoplasmic reticulum, the pre-proinsulin. In the Golgi apparatus, it is scissored off here so that this part is available separately as insulin, active insulin, which is from the total 51 amino acids. 21 amino acid insulin is formed and 30 amino acid C peptide remains. And that is what you measure indirectly if you want to measure the insulin. The C peptide levels correlate with the insulin breakdown and produced in the Golgi apparatus. Okay. Now, beta cell, the KATP channels are there, and then the glucose calcium channels are there, we know, and the glucose hyperglycemia is there. It goes inside, closes the KATP, opens the calcium, releases insulin. We all know that. KATP sulfonylurea receptor is very important. There is what is called the SUR receptor, sulfonylurea receptor. These receptors are on the beta cells and also on the cardiac muscle. So the sulfonylurea receptor, in fact, facilitates the closure of the KATP channels when the sulfonylureas are there. When there is glucose is not able to stimulate enough insulin, the sulfonylurea goes and sits on this receptor and closes the KATP channel. That's where sulfonylurea acts, and then this automatically opens the calcium channel and releases insulin. This is the mechanism of action of sulfonylureas. Normally, the process is through the glucose, closing of this door and opening of this door. But sulfonylureas being secretagogues, they have a receptor, their SUR receptors. They close the KATP channels. With the result, calcium channels are open and then insulin is released. This is the mechanism of action of sulfonylureas. KATP channel has two subunits, KIR 6.2 and the regulatory part. This is the SUR part and the KIR 6.2 part. The regulatory part is the SUR part. ATP-gated potassium channel is coupled with SUR. That is the SUR receptor, sulfonylurea receptor KCNJ11 gene, and KCNJ11 gene encodes TCF762 via proglucagon gene expression or GLP-1. That is how GLP-1s are stimulatory at the beta cell. K channel can be closed independent of glucose. We are not asking glucose to do this job. Instead, we have employed a person to close the KATP channels, and that is either GLP-1 or sulfonylurea. What does this lead to? It opens calcium channels and then releases insulin. SUR or ATP binding transporter superfamily. So SUR receptors are present in the muscle, cardiac muscle, and also on the beta cell. That is why the long-acting or older generation sulfonylureas are cardiotoxic because of the SUR receptors being present on the cardiac muscle. So what happens? How does the insulin get secreted? Insulin, how does it allow the movement of glucose into the cell? Earlier, we have seen how insulin comes out, and having had the insulin, how does the insulin take the glucose into the cell for metabolism? Because glucose, unless it enters the cell, any peripheral cell like muscle or adipose tissue or any other cell for metabolism, it will be lurking in the blood and causes hyperglycemia. So how does it do? When the insulin, this is the tyrosine kinase receptor, it has got two gates which are wide open. Now, when the insulin molecule is there, they become together. They come together and bind the insulin. This is the cytoplasmic, this is the extracellular fluid. And once the insulin is there, They bind to the tyrosine kinase receptor and they come together and pick up the insulin molecule. Once the insulin molecule is here, what it triggers is several chain reactions occur through these two limbs, and this ATP is converted into ADP and phosphate is released, and phosphate is added onto the terminal of the tyrosine kinase. So this is now became phosphorylated tyrosine kinase. Phosphorylation is the signal. The moment phosphorylation occurs, tyrosine kinase is phosphorylated, now active, a cascade of effects that leads to allow entry of glucose and any anabolic activity. So what is happening? Insulin comes into the blood from the pancreatic beta cell. That insulin goes to the effector cell, the adipose tissue, or the muscle cell mostly, or any other cell, and that goes and touches the tyrosine kinase receptor. Those receptors, the two halves are far away, and once they see the insulin molecule, they bind to the receptor proteins there, and that results in cytoplasm dimerization. That means the two receptors, the two halves come together and pick up the insulin there. Once the insulin is there in the receptor, it triggers a series of changes in the limbs of the two halves of the receptor. With the result, that requires ATP to form. ATP at the end, and the available energy, phosphate, is bound to the terminal end of the receptor. That is here. And that is what is called activated tyrosine kinase. And there can be mutations where tyrosine kinase is not properly activated, then insulin will fail to do its job. Or insulin has done it, but there is problem in the cell, the phosphorylation is not occurring, tyrosine kinase is not activated, and then this glucose and other glucose uptake and other things have not come. Insulin receptor is tyrosine kinase, consists of two subunits, and they dimerize when bound with insulin. Inside the cell, autophosphorylation occurs, increasing tyrosine kinase activity. IR phosphorylates insulin in the intracellular signaling molecules and then stimulates insertion of GLUT4 protein to let glucose in. All this leads to GLUT4s are inside the cell, and the GLUT4 doors are opened outside the cell. So GLUT4 now is a channel to enter. They allow the glucose to enter into the cell for metabolism. For this, what is required? Tyrosine kinase should be phosphorylated. For that, what is required? Insulin is required. So this is called the insulin signaling pathway. Through this, what happens? GLUT4s are open onto the cell surface, and glucose freely enters to the GLUT4. Whereas there we have seen GLUT2 in the beta cell, that doesn't require insulin. In fact, GLUT2 allows glucose to go inside and produce insulin. Here, insulin allows glucose to enter into the cell through the GLUT4. They stimulate glycogen, fat, and protein synthesis. Also, if excess glucose is there in the cell, it goes into glycogen or fat or protein. So this is here you see the alpha unit and the beta subunit at the membrane level. This is the tyrosine kinase domain, and the C-terminal tail is there, and the insulin receptor is there. And once the insulin is sitting here, and then this is phosphorylated. That is what we have seen there. So this is one unit. These are the both units unphosphorylated, now completely phosphorylated, active tyrosine kinase phosphate. Similar in structure is epidermal growth factor receptor. EGFR is the same as in structure. It is like a tyrosine kinase receptor. What is the use of this? If I have a drug which can stimulate the EGF receptors, it will act like insulin and then produce a secretagogue effect, which is a novel secretagogue. Epidermal growth factor receptor, which just mimics the tyrosine kinase, which is stimulated by the insulin. Instead of insulin, epidermal growth factor can do that, and this is the same mechanism there. Instead of insulin sitting here, epidermal growth factor sits there, and if I have a drug which stimulates the epidermal growth factor receptors, there is autophosphorylation of tyrosine kinase, a novel secretagogue, which is now being very being evaluated. We will have soon a drug which will be a novel secretagogue. Now, this all of you have seen earlier. So, in the initial stages, there is no hyperglycemia. This is fasting for postprandial. This is the fasting is the first one to occur at the time of diagnosis. Now, initially there is increased insulin secretion to start with, and that comes down. This is apoptosis of beta cells. This is insulin resistance. So, what will be my first feature? Fasting hyperglycemia, followed by postprandial hyperglycemia, and then hyperinsulinemia, followed by hypoinsulinemia and insulin resistance. This is the time course. If 0 is the diagnosis, the damage starts 20 years earlier and continues for 30-40 years. So this is the relative beta cell function. The relative beta cell function at diagnosis is 50% of the original capacity of the beta cell. So what is this? This tells me that I can protect the beta cell well before the diagnosis of diabetes. That is what is called treatment and management of pre-diabetes. So what does happen? There is insulin resistance and there is decreased beta cell function. Blood glucose increases, insulin is a response occurs, there is an adequate response, euglycemia, inadequate response, type 2 diabetes occurs. What does this cause? What is this caused by? Obesity, sedentary lifestyle, aging, genetics, glucotoxicity, free fatty acids, lipotoxicity, all act on beta cell function, and they decrease the beta cell function and increase the insulin resistance. And these speak with one another. Decrease the beta cell function, in turn promotes insulin resistance, in turn releases and decreases the beta cell function. Now I have several mechanisms to control diabetes: decrease obesity, slow carbohydrate digestion by your alpha glucosidase blockers like acarbose and oglibose, slow the gastric emptying by now prokinetic drugs, alter glucose fatty acid cycle, stimulate glucose utilization by sulfamin by the thiazolidines and metformin, increase the glucose excretion, SGLT, suppress glucose production from that is metformin, and then from the glycogen reduction into glucose is reduced, decrease counter-regulatory hormones, that is glucagon suppression, insulin replacement is not there, increase the beta cell mass, increase the insulin secretion, and improve insulin action. These are several ways you can address the glucose. Normally, we are targeting one or two. One is this is there, and then this is also used, and then this is used, and then this is used. But every mechanism can be tried to bring the blood glucose into control. Approaches to lower the blood glucose. Now, I am skipping on the left part of the slide and right part of the slide. You see the free fatty acids here. Free fatty acids are inflammatory. They increase the oxidative stress. They increase the endoplasmic stress. There is autophagy of the beta cell apoptosis. This is the negative aspect of free fatty acids. And then beta cell function is decreased. Here, the positive aspect is free fatty acids. They facilitate the beta cell function through the PPARs. Beta cell survival is increased, beta cell proliferation, cell regulators, growth factors, incretins, all act. It is a two-way sword, two-edged sword. If it in the normal circumstance, it protects the beta cells and makes them function. If the free fatty acids are more outnumbered, like lipotoxicity, then the red part of the series comes, and then beta cell apoptosis. And this is very simple also. Krebs cycle here, the pyruvate, and then the free fatty acid cycle. Free fatty acids are metabolized like this, glucose is metabolized like this, and then this is again the free fatty acids in a different pathway. They go to the Krebs cycle, and ultimately all these have a bearing on the insulin release and the calcium channel opening and things like that. Okay, now net beta cell mass. Net beta cell mass in the pancreas depends upon new beta cells that are formed and number of beta cells that die. Die means not structurally but functionally. Apoptosis is functional death. Structural death is necrosis. Apoptosis is functional death. Cell is alive, but it is not functioning. That is apoptosis. You understand the term apoptosis. Apoptosis is the cellular organelle are intact, but functionally they are not doing their function. That is called apoptosis. Necrosis, cell death. Okay. And then there is replication happening. So the factor that decides the beta cell mass is how many new cells are formed, how many are dead. That is the net beta cell. So either I can increase the neoformation or I can prevent the apoptosis to increase my beta cell mass. That is the emphasis here. The formula of net beta cell mass is mitogenesis plus size plus neogenesis minus apoptosis is the growth. That is generally everywhere. What is the amount of mitogenesis? What is the size of the mitogenesis? And then the neogenesis, new, not from the old ones, new cells forming. And that together is the input minus the output, the apoptosis, which is the growth. Increased beta cell mass, compensation for insulin resistance, because when the body is having insulin resistance, automatically insulin by increasing the beta cell mass occurs. Mitogenesis plus size plus neogenesis is more than the apoptosis. It is taken care of. Other than that, my apoptosis is more than the formation. Then there is a decrease in the beta cell mass, which leads to type 2 diabetes. This is a very tricky balance between the harmonious maintenance of beta cell mass normally or decreased beta cell mass. Now, this is genetics as well as environment. Both are involved here, and you have insulin resistance under the genetics as well as the environment. Genetics and environment, and they trigger the normal beta cell function, compensatory hyperinsulinemia, and no diabetes. Suppose there is an abnormal beta cell function coupled with insulin resistance, that leads to relative insulin deficiency, which is called the type 2 diabetes. And the balance is what makes a person non-diabetic into a diabetic or pre-diabetic. What are the factors? Genetic factors, environmental factors, beta cell factors. All the three are important. Now you see there are two phenotypes, the lean phenotype and the obese phenotype. If a person is a lean phenotype, there is impaired glucose tolerance, there is impaired fasting glucose, and that translates into oxidative stress and glucotoxicity, cellular lipid synthesis, and glucolipotoxicity, progressive beta cell failure, and type 2 diabetes. Okay, there is an obese phenotype. These have elevated free fatty acids and initial glucolipo adaptation, that is the greener part of that particular slide, increase the FFA usage, and that produces hyperglycemia later, glucolipotoxicity, ending in beta cell failure and type 2 diabetes. Either way, the lean type will not have elevated free fatty acids. It is mainly hyperglycemia, either for postprandial or fasting, that drives the car. Whereas when somebody is obese, it is not important that the glucotoxicity is more, but the lipid also comes into picture. Together they cause the problem. Now, beta cell failure can occur glucotoxicity, lipotoxicity, ER stress, mitochondrial dysfunction, oxidative stress, islet cell inflammation, increased amyloid polypeptide, which we have discussed in this paper. All of these have a chain reaction. They increase the insulin resistance, and against the genetic background of insulin resistance, that leads to beta cell failure. What are all they? Glucotoxicity, lipotoxicity, ER stress, mitochondrial dysfunction, oxidative stress, islet cell inflammation, and increased islet cell amyloid deposit. All have a sequential chain reaction under a genetic background that leads to insulin resistance and type 2 diabetes and beta cell apoptosis. Effects on multiple organs. So for example, there is hyperglycemia, there is malonyl coenzyemia is more, fatty acid coenzyemia is more, hyperlipidemia. So there is a hyperlipidemia, fatty acid coenzyemia, malonyl coenzyemia. Hyperglycemia also produces malonyl coenzyemia. All have an effect on the muscle, on the adipose tissue, on the beta cell, on the blood vessel, on the neural, and on the liver. See how bad it is. On the adipose tissue, increased adipose tissue mass, obesity. On the beta cell, altered secretion and cell death, diabetes, insulin secretion is decreased and beta cell apoptosis. On the vascular, altered blood flow and contractility, and then atherome proatheromatosis state. Diabetes related complications on the nerves, DPN, autonomic DAN, that is diabetic autonomic neuropathy and peripheral neuropathy. And on the liver, excessive glucose production, fatty liver, and insulin resistance on the muscles. You see, the same hyperglycemia and hyperlipidemia, the glucolipotoxicity together has got multiple effects on multiple organs, as can be seen here. Now, this picture is from the same article which we have given. Once there is sustained hyperglycemia, hyperlipidemia, that is glucolipotoxicity, and PGC1 alpha, PPAR coactivator, PPAR gamma coactivator, PGC1 alpha is increased. That results in beta cell function there and decrease the insulin secretion. Together, they are increasing the PGC1 alpha, which is an inhibitor of the beta cell and beta cell function, and then the insulin is decreased. Glucolipotoxicity, saturated fatty acid, palmitate is harmful, oleates protective, short-term, long-term exposure, impaired insulin gene transcription, glucose-induced, glucose-stimulated insulin secretion, pro-apoptotic and anti-apoptotic. Oxidized LDL enters the beta cell, ABCA1 disruption, increase in the islet cell cholesterol, impaired GSIS, impaired fasting glucose and IGT, LXR antagonist, ABC activation is the answer. Now, what are we saying? When we have increased glycemia, increased lipidemia, not only this is the mechanism of reducing the insulin and causing glucose-stimulated insulin release reduced, it also thinks about oxidized LDL. There is OX-LDL. Oxidized LDL will enter the cell. Normally, when the oxidized LDL enters the cell, it is taken away by the scavenger receptor, ABCA, and pulls it back and throws it into the liver and dumps in the bile and excretes the excess LDL. What happens in this case when there is glucolipotoxicity? Whatever OX-LDL enters here, this is because the ABCA1 is disrupted, this OX-LDL is no more moved out of the cell and it stays there inside the cell. That means there is a lipid deposition or a cholesterol deposition, LDL deposition within the cell. That is called intra-islet cell cholesterol deposition. What does it do? It impairs the GSIS. It impairs the mechanism of GSIS and potentiates the diabetes. How do I counter that? I use ABCA1 LXR antagonist, and which will block the LXR receptor and activate the ABCA1. ABCA1 is a scavenger pathway. It takes away the OX-LDL. And so this is one mechanism. We can help the GSIS by removing the effect of low ABCA1 by using LXR antagonists. This is one treatment option. Now, mechanism of beta cell failure. The beta cell is a central cell there. There is glucotoxicity, there is amyloid deposition, there is oxidative stress, there is endoplasmic stress, there is inflammation, there is lipotoxicity, which we have seen in the previous slide. All together, apoptosis and reduction in the beta cell mass. Now, glucolipotoxicity on the beta cell, there excess free fatty acids are there. Remember, free fatty acid is one of the very powerful biochemical molecule in the body with several adverse effects. It is worse than many molecules. The moment you have free fatty acids more in the system, insulin resistance is triggered, beta cell apoptosis is triggered, several endocrine functions are hampered, and then fatty liver occurs, and fatty deposition in the pancreas is today's watchword. Earlier, we were thinking of fatty liver. Now we do an ultrasound and we see a fatty pancreas. How much fat is there in the pancreas will decide the future of the individual. This produces ceramides and PKC, and these increase the endoplasmic stress and oxidative stress, and also produce inflammatory cytokines, IKKB and JNK activation. So this say inflammatory cytokines on one hand, oxidative stress and endoplasmic stress on the other hand, they speak to each other, they interact and potentiate impaired signaling, insulin signaling in the beta cell. When insulin is not signaled, it is not allowing the glut to open. So glucose is circulating in the blood, not allowed to come into the cell for metabolism. Insulin resistance occurs. Hyperglycemia prevails. Hyperglycemia triggers more insulin secretion from the beta cell because we know glucose enters the cell and produces. Ultimately, from the bank, more is drawn, and ultimately, bankwala will say, "I don't have any more deposit of you. Everything you have exhausted." And the beta cell apoptosis occurs. See now, free fatty acids, ceramides, oxidative stress, endoplasmic reticulum stress on one side, inflammation through the cytokine mediated. These two together will impair the insulin signaling. We know insulin signaling is essential. What is insulin signaling? Phosphorylation of tyrosine to form active tyrosine kinase. And that is missing. Glut is not opened onto the cell surface. When the GLUT4 is not opened, glucose is not entering into the cell for metabolism and utilization. Glucose stays in the blood, hyperglycemia continues. This hyperglycemia doesn't keep quiet. It goes to the beta cell. Oh, we are so many molecules. Glucose here, you close the potassium doors and open the calcium doors and release the insulin. They go and press the insulin releasing mechanism in the beta cell. Then what happens? Beta cell will try to give hyperinsulinemia to start with for some time. Then it says, no, I don't have any more money left. Supposing mother has 1,000 rupees. The boy goes on asking and pressing her to give money, 5, 10 like that. At some stage, mother says, I don't have any more money. All the money I have already given to you. Thank you. Don't come to me. So that's what the beta cell will say. That is what is called beta cell apoptosis. How do I prevent that? I prevent fatty acid accumulation, or I prevent the inflammation, or I tackle the endoplasmic reticulum stress, and then prevent beta cell failure. These are the newer mechanisms of treatment. Now you see, this clearly explains when glucose is there, through the GLUT4 it comes, and insulin is facilitating that. This is an activated tyrosine kinase receptor, and then decrease in the PI13K, which is also a very important molecule for beta cell function. In the presence of excess plasma fatty acids, lipoprotein lipase, and then the free fatty acid receptors are there, fat P1 receptors, the free fatty acids enter, fatty acid acetyl coenzyme A, diacetylglycerol is stimulated, mitochondria are stressed, and this increases, this in turn also increases the inflammatory JNK1 and IIKB, and then IRS1 serine phosphorylation occurs. Insulin phosphorylation, tyrosine phosphorylation is reduced. Insulin is no more able to open the glut, and hyperglycemia prevails. This is the biochemistry behind what I have described in the previous slide. Okay, this is again from the article here. Normally glucose oxidation, detoxification, insulin granules are released, insulin secretion. Beta oxidation, lipid detoxification, together they promote insulin secretion at a normal level. But if this is more, and if this is also more than it can handle, then there's glucolipotoxicity, then the hyperlipidemia, long chain fatty acid acetyl coenzyme A, decrease in the oxidation of them, increase in the glucose detoxification is decreased, glucose is not detoxified, oxidative stress occurs. Now the hyperglycemia produces more of malonyl coenzyme A, decreases the PPAR, which are protective. This decreases the beta oxidation of fatty acids. Lipid detoxification is decreased. Here glucose detoxification is decreased, and lipid detoxification is decreased. That means glucose and lipid together are now toxic to the cell. What do? They increase the central molecule, diacetylglycerol, phospholipids, and triglycerides. This diacetylglycerol, when it is increased, it increases oxidative stress. It produces ceramides. Ceramides disrupt beta cell function. That is beta cell apoptosis. That is worsening of type 2 diabetes, and then the end result is micro and macrovascular complications. Now, what are the stages? Rajaram described to some extent. These stages. So these stages, there are three different stages. Stage of mild hyperglycemia. Please close your mic. Mild hyperglycemia and severe hyperglycemia and decompensated stage. Now, this is the today's slide is this one very important one. TNFR5 STAT1 NFKB pathway mediated glucolipotoxicity. TNFR1 human necrosis factor receptor 5 signal transducer activator of transcription STAT1 and NFKB nuclear factor kappa B are the three mediated. These are the three mechanisms through which glucolipotoxicity is mediated. What does it do? DNA replication, 4% of it, cell cycle, metabolism, telomere maintenance. I think you must be knowing that the length of the telomere decides how much long we live. Today we can measure exactly, almost to the precision of a few days, how long a person will live, depending on the telomere length. If you know the telomere length, you can know the longevity. How do I keep the telomere longer enough? By reducing my sugars, by reducing my lipids, by reducing my hypertension. All these are possible by diet and exercise. So people who live longer are because genetically, of course, the telomere size is important, but environmentally, telomeres can be maintained by positive lifestyle. Okay, and diabetic pathways, signaling pathways, apoptosis, biological oxidation, gene expression, RNA processing, axon guidance in the neurons, homeostasis, DNA repair. These are the various functions of TNFR5 STAT1 NFKB mediated glucolipotoxicity. When you teach postgraduates in your near future, you will be having drugs which will affect these things and positively control glucolipotoxicity and favorably maintain all these functions. This can be accessed at www.reactome.org. Reactome is a very experimental group, very strong experimental group working on this model. And you can see there are 13 significant biological functions as shown in this slide are identified. Now I am going to, left side I will see increases free fatty acids, reduced adiponectin, which is a protective substance, increased leptin, which is a dangerous substance, produces adiposity, reduces adiposity. Adiponectin reduces adiposity. Leptin increases the adiposity. And these increase the demand for insulin, elevated cytokines, chemokines, and then the lipooxygenase 12 also. Excessive intake of calories, adipose tissue, and that produces all these things. And then adipocyte hypertrophy, hyperplasia, and these act on the immune cell recruitment also, and CD4s are also affected there. Adipocyte inflammation occurs, insulin resistance, type 2 diabetes. So what is that? Reduce excess calorie intake and increase the exercise and don't allow the body to accumulate excess fat. The left side, I'm going to share the presentation. You see, in nutshell, it is oxidative stress, decreased oxidation of fatty acids, decreased PDX expression, decreased insulin secretion, and result is glucolipotoxicity. Now I'm coming to the end of the presentation. Both in type 1 and type 2 diabetes, beta cell dysfunction is the most important thing. It can be autoimmune through HLA mediated because it depends on the HLA sequence of your body, how much beta cell apoptosis occurs and how fast it occurs. It is autoimmune mediated. Now we know type 1 diabetes is of course autoimmune disease, and type 2 diabetes also has got several HLA mediated autoimmune phenomena. Insulin resistance through this KCN-J11 is triggered. Adipokines, cytokines, viral infections, other insults, beta cell dysfunction, decreased insulin secretion, glucotoxicity, lipotoxicity on the beta cells, oxidative stress, endoplasmic stress, inflammatory cytokines, impaired self-repair. You know the beta cell mass is replication minus the apoptosis. New cells formed now is disrupted and cell damage and apoptosis occurs. And how do I do that? by alleviating the beta cell stress. The green one shows there is a way out for that. Here, this is pancreas, healthy, genetically susceptible beta cells, adipokines, viral infections, other insects. They change the beta cell into a sort of toxic beta cell, glucotoxicity, oxidative stress, pro-inflammatory cytokines, interleukin 1 receptor antagonist. With no intervention, this goes to apoptosis and cell death. With intervention, it goes to the green side, improved insulin secretion. Now we have interleukin 1 receptor antagonist, which is anakinra, which we'll see in the next slide to tackle the glucolipotoxicity. Regulation of beta cell mass, a dynamic function, which we know that neogenesis, replication, hypertrophy, increased beta cell mass, decreased beta cell mass, apoptosis, necrosis. This is the end result. The expression of interleukin 1 receptor antagonist is reduced in the pancreatic cells of patients with type 2 diabetes mellitus. High glucose concentration induce the production of interleukin 1b in human pancreatic beta cells, leading to impaired insulin secretion, decreased proliferation, and apoptosis. Decreased proliferation means no beta cells. Increased apoptosis means beta cells are lost. So net result is negative glucose metabolism balance. Now you see what happens. You see here numerous factors influencing type 1 and type 2 diabetes susceptibility, genetic variation, environmental exposures, maternal influences, aging, cellular senescence, all have a role on the pancreas, on the islet, on the apoptotic beta cell, loss of beta cell, insulin secretion, and beta cell mass, diabetes susceptibility. Homeostatic balance between protein production and insulin secretion, healthy beta cell, normal pulsatile insulin secretion, diabetes is prevented. So it's a question of whether you have a healthy or unhealthy. It depends upon the various factors that are acting on that. Now you see. So decreased interleukin 1 receptor antagonist means what? This expression is reduced IL-1A, IL-1RA, increased glucose IL-1B, counter-regulator is increased. This triggers decreased insulin secretion and decreased cellular regeneration, apoptosis. What is this? These are chemokine mediated or cytokine mediated. If I have a drug which can interfere with this reduction, and I can stop the whole cycle. That is whole story is that. So the anakinra versus placebo, and 312 patients are recruited, 108 did not respond, 124 were screened, 54 were not eligible, 70 underwent randomization, 1 excluded, 34 were assigned to anakinra and 35 to placebo, and then 34 completed study, 33 completed study. What is the beauty? This is placebo, this is anakinra. So the glycated hemoglobin reduction is very profound with anakinra. As you can see, this is the placebo and this is the anakinra. That means, hemoglobin A1C reduction is very much in glycated hemoglobin, which is HbA1c. So this is again, the various dose body surface area, depending on how obese they are. The less obese, the more effective it is. That also is seen. Okay. Now, this study suggests that antagonism of interleukin 1 has a possible therapeutic potential in the treatment of type 2 diabetes. Anakinra needs to be evaluated for its long-term use and preventing beta cell destruction and promoting beta cell regeneration. This is what is called bridging the gap. This anakinra is approved as early as the turn of this century for rheumatoid arthritis. Anakinra, a recombinant version of natural interleukin 1 receptor antagonist, blocks interleukin 1 alpha and 1 beta, FDA approved in 2001 for rheumatoid arthritis, improves glucose control, increases insulin secretion, decreases inflammatory markers, multiple effects. TNF blockers like adalimumab, cetulimumab pegol, etanercept, golimumab, infliximab, gamma interferon, and glycogen synthetase kinase 3B inhibitor, and histone deacetylase inhibitor. All are under trial. What are these? Anakinra is already tested and will come into the market. All the other are the cytokine blockers. What these do? They prevent the interleukin 1 receptor antagonist blockers. And we have seen that mechanism protects the beta cell. Now, here is an answer. Instead of trying to treat diabetes once the dysglycemia occurs by secretagogues, by uh, drugs which will increase the insulin utilization or by insulin itself, here is a mechanism I can use to prevent beta cell apoptosis and see that diabetes doesn't develop. So these are the ways and things. So there are several papers, diabetes treatment, bridging the divide. This is the title of my talk based on most of these papers. Insulin interleukin 1 receptor antagonist and type 2 diabetes, glucolipotoxicity and pancreatic cell glucolipotoxicity. This is the article which Rajaram reviewed. So there are several articles like that which are now promising the right direction. Not that they're ready to be used tomorrow, but these will be tomorrow.